Okay, recording is in progress. So just so you all know, um, and it'll be posted on the ICC website and on YouTube with captions so that you can revisit it or you can share it with your colleagues who weren't able to make it today. Okay, that is all from me. Chris, you can go ahead and share your screen and take it away.
Okay, can everybody hear me okay?
Yep, we can hear you, and we're
Okay,
seeing your
great.
presentation too.
All right. Everybody seeing the screen? Of course, I start my presentation and I'm getting a spam call at the same time, right as we start. Okay, thank you, Natalia. And welcome everyone. Looking over the list of participants, there is an awful lot of crayfish expertise on here. Some of the folks probably know more than I do, especially when I call out one specific person, Premic Hammer from Canada, is an expert, a world expert on one of the species I'm going to mention. So a great group of people. Um, and as Natalia mentioned, if you have questions anytime, I get rolling and talking and I'm not paying attention to that. So Natalia, I'll rely on you to just interrupt me at any time if there's a question on the chat. Okay, so to start, um, this presentation is a portion of a one-day workshop that I co-developed with a close colleague of mine, Gunter Schuster. So a lot of the material, some of the slides, and an overwhelming majority of the photographs are Gunter, so I definitely want to acknowledge him and Susan Adams from the U.S. Forest Service for sharing some of her photos for this. So with that, we will begin. Um, just to back out from the Midwest initially to get our bearings on crayfishes in general. The point I want to make with this slide is that crayfishes are, I'll say, fairly old invertebrate group. Um, DNA evidence from over 25 years ago shows a monophyletic group of freshwater crayfishes that predate the split of Pangea into the two supercontinents. So we can assume from this data that the ancestors of freshwater crayfish invaded freshwater prior to the split, which is somewhere plus or minus 200 million years ago. So for an invertebrate group, fairly old compared to vertebrates like fishes at 400 or 450 million years, not that old. But remember, bones preserve a lot better than exoskeletons, so we may be missing some signal on age for crayfish. Okay, just a rough overview of crayfish's distribution in the world. This is a figure from Hobbs that goes way back with some updated numbers of diversity. So we're, over 600 species and subspecies of freshwater crayfish. Um, there are two major groups going back a slide. There is the peristachids, which are the southern hemisphere crayfishes here at the top, and the northern hemisphere crayfishes that are composed of two families, the Astacidae and the Camberidae. This is a distribution of diversity across those two groups, the northern and southern hemisphere families of crayfish. Um, Just an example of the southern hemisphere crayfishes. This has absolutely nothing to do with Midwestern crayfish biology or ecology. I'm going to admit that straight up. But this is probably the coolest freshwater invertebrate and the largest freshwater invertebrate in the world. It's a member of that southern hemisphere family, and I love an opportunity to throw a slide like this in any talk. So Premic Hammer from Canada did his PhD dissertation on this species and probably knows more about it than anyone, and he's on this webinar today. So, anyways, all right. So let's get to what we're going to talk about today. This is just a quick outline. We're going to cover three basic sections: life cycle of Midwestern crayfish, biology / life history of Midwestern crayfishes, and then touch a little bit on ecology. But looking at the names of the list or the people that are on this webinar, most of the ecology section is going to be very old hat. I'm not going to have anything new for those folks, but we'll touch on it at the end. All right. So we'll start with life cycle. Very simple life cycle for freshwater crayfishes. We start with an egg. There's an immature stage because there is direct development in crayfishes. We'll circle back to that. Immatures develop into adult female and adult male. The adult male is where things get a little bit more, I'm not going to say, I don't want to say complicated, but a little bit more abnormal in that there's two forms, form one and form two, and that is only in the family Camberidae. If you remember from the map, all crayfishes in the eastern U.S. and Midwest are members of that family. Okay, so just kind of the main difference. If you remember back to that phylogeny slide, I didn't point it out, but the closest known relative of freshwater crayfishes are marine lobsters. And in marine decapods, like most marine organisms, including fishes, there are larval stages. Look at the two images at the top of this slide. You'll see what a larval spiny lobster, Caribbean spiny lobster, looks like. It doesn't look anything like an adult. Crayfishes, on the other hand, with that direct development, this is the main difference, even though crayfishes and lobsters are in the same order. They're all decapods. Freshwater crayfish have direct development. And by that we mean juvenile or immature crayfishes that are attached to the underside of females. You could look at them and with a very small amount of experience know that they're probably juvenile or immature crayfishes. So that direct development again is the main difference between the nearest relative, the clawed lobsters, marine lobsters. All right, so any talk about biology or reproduction in Canberra crayfishes here in the Midwest very quickly comes to form and form alteration between form one and form two. And we're going to get into that in the next few slides. There are some marked morphological differences, but the difference I want to show with this slide is that form one males are generally a little bit larger than fully mature females of the same age. And some of the morphological features of form one males do become larger. Notable here in this slide is this size of the chelae on this form one male. The size of the chelae in form one, even versus form two males, can be different. So those form one males have more pronounced and large morphological features, in addition to generally having a larger body size. Okay. Sexually dimorphic crayfishes, obviously, most of you already know that. Just as a quick image of how males differ from females. The males on the left, I'm sorry, have two modified sperm transfer organs that we call gonopods, as you can see here. Females, on the other hand, lack those structures and have a sperm storage organ called the annulus ventralis. Also want to point out on this really good photo from Susie Adams are the glare glands. We're going to talk about that in an upcoming slide. You'll see these creamy white patches that are just subcutaneous in female crayfishes. Okay. Female crayfishes get these glare glands as they get close to laying their eggs. They don't have them all the time. All right. So as I mentioned on the previous slide, this structure on female crayfish is a sperm storage organ. This is not an organ that releases gametes or eggs, unfertilized eggs. Those in the female crayfish come from a gonopore that's on the base of the third pair of walking legs, as you can see from the arrow. It's kind of covered with CD here in this image, so it's not showing up real well, but there is an opening that's covered with tissue right under this CD here at the base of these walking legs on each side. Okay. Males, on the other hand, have the sperm transfer organs. And these photos of Genters are very good at showing differences where in the structures between form 1 and form 2. Okay. Form 2 males are the reproductively inactive form here in the top right. Form 1 are the reproductively active form on the bottom right. And the first difference you should notice is the difference in detail. The Form 1 gonopods became way more, well, they become thinner. Um, they get a yellowish tint, we call that cornified at the top edge of the structure. They become more acutely tipped and just in general more detailed. Okay. Sometimes you'll hear the male gonopods referred to as the first pleopods. Pleopods are the 5 pairs of what are also called swimmerets on the underside of the abdomen. This would be the second pair that's shown here. The first pair are kind of buried under these second pair. So sometimes if you're looking at genitalia in crayfish, you have to be careful that you're not confusing the first pleopods or the gonopods with the second pair, which will overlay them on top. So you've got to remove that second pair or pull that second pair back to see the first pair. All right. An important note here is that the gonopods are solid structures. There's no internal tube that moves the sperm down that gonopore. It relies on just the movement of the spermatozoa along a groove that's on the outside of that gonopod. And you can see that groove marked here on 3 different genera of crayfishes. It's always there. And so the spermatozoa are released from a pore at the base of the 5th pair of walking legs in males. Okay. So again, it's on the 3rd pair on the females. Up here in the males, it's on the 5th pair. And the spermatozoa are released and they just come in contact with these gonopods and just slide down the grooves on those gonopods, wrap around the structures called the mesial process and down the tip for transfer to the females. Okay, getting into the life cycle now. This is a photograph or a figure that a lot of you have probably seen in other publications or other talks. It's very hard to follow. I wish they would have contrasted the color of this area of the figure with the area in between these color changing bar or bands. Okay. What I'm trying to show, so this is a generalized Canberra life cycle, and it fits pretty good for Midwestern species. And we're looking at, the 12 months of the year. And this is the form alteration, where Form 1 males are shown in the shaded gray, and the open white pathway are for Form 2s. And what I want you to take out of this is that generally, as crayfish age, there's a spring molt and a full fall molt. And we're talking about molt now because males molt into the different forms. There's a molt from Form 2, as you can see here, through the summer months into the Form 1 reproductively mature stage, which they stay at through most of the winter and early spring months. And then you get into spring and you molt out of the Form 1 stage into the Form 2 stage. Put some cooler photographs to that life history. again, generalized for Camberdae, especially in the Midwest. If we start here in late winter or spring, the female secretes, fertilizes, and incubates her eggs. Okay, we'll get into that in detail in the next few slides. A lot of you have seen pictures of ovigerous or females in berry that carry their eggs that have been fertilized on their underside. Those eggs hatch and develop. Again, we'll get into detail of that in a second. The juveniles are released by the females. You see a period of rapid growth through the summer months and then go into the mating phase in autumn. Okay. What I want to point out here is that age 0 males can mate. So that means that in that very first fall of existence, if they were the juvenile in spring, they went through a period of rapid growth, very, very small at age 0, but they still can molt into a reproductively mature Form 1 male that first fall. How many successful matings do they have at that small size? I can't say, but they are able to transfer gametes. Okay, so that's the basic life reproductive strategy and life cycle. I'm just going to go into detail now on these next few sections of biology. Any questions? Take a quick break. If I don't see any message from you, Natalia, I think we're okay then.
Yep, we're okay so far.
Okay, all right. So I'm going to talk about each of these here in the preceding slides. Courtship behavior, um, copulation, storage, fertilization, and berry and egg hatching. All right, so getting into crayfish sex here. Again, this is generalized because of the 400 plus species in North America. Detailed life history or reproductive studies have been conducted on probably less than 5. So what we think we know might work for the majority of the species, but there most definitely can be some exceptions to that. So again, generalized description of this copulation interaction. And a lot of this was done in laboratory studies. I would say all of it has been done in laboratory studies. As you can tell by the very authentic looking gravel here in the background on these photographs. Generally, the copulation starts with tactile interactions with antennae. There's some like touching between the two individuals. There's a lot of data that shows that there's sensory perception between individuals also before it gets to this tactile interaction. Males may be able to sense females or vice versa from urine that's released by individuals. So there's probably some interaction before this tactile interaction that's happening in a natural environment. But most times you'll see this light touching with antennae. It may be a form of gender or form confirmation. It's really unknown exactly what is going on with that touching. It's just normally a component of this courtship behavior. Male is usually initially aggressive, more so than the female, and the female usually initially resists that initial advance. After minutes or could even be an hour or more of this interaction with touching and again, surely some smell in the water, the female will eventually relax and submit and stop moving their chelae and walking legs and will basically just kind of lay there limp. And then the male will move into position, flip her over on her back using his chelae and ischial hooks. I have a photo of that coming up in the next slide. And then we have gamete transfer. Okay, well, some of you may have seen this interaction in buckets in the field. Sometimes if you're out at the right time in the fall or early spring and you put males and females of the same sex in a bucket, if you leave them alone, 20 minutes later, you'll go back and look in the bucket and you'll see you'll see the activities going on. All right, so the ischial hooks, that's another difference between males and females in cambariid crayfishes. Looking at the structure here in the third pair of walking legs or pariapods. These are structures that are usually generally used to lock the female's legs outward and kind of firm up that bond for gamete transfer going on by the male gonopod. All right. Again, a handful of studies that have looked at this, but we do know that females will molt with multiple males. The males will use a plug, a sticky plug, to cover that annulus opening after they've done mating, but there's also evidence to show that then another male will come along and remove that plug and transfer sperm. That sperm is being transferred to the annulus ventralis, as I mentioned in that earlier slide. That is just a sperm storage organ, okay? We're having no fertilization of the eggs going on at this point. Gametes are still held separately. There is some evidence to show that, again, I mentioned chemical signaling may play a large role, and that males may be more receptive to virgin females that haven't already mated. Here's the really interesting aspect, and that is, again, so that sperm is transferred into that annulus ventralis. Females can retain that viable sperm for months. And so remember from that slide, that protracted spawning period or copulation period can start in the fall, and egg laying normally happens in the spring months. Those females are holding that viable sperm in that annulus ventralis for sometimes 6 or 8 months. So a pretty unique feature to maintain viable sperm for that period of time. Probably a bet hedging strategy, I would guess, on why crayfish do that. Allows them to have many more opportunities to mate and store that sperm until the following spring. In case you have an extremely dry winter or cold winter or long winter, drought conditions in the spring, it seems like an excellent bet hedging strategy. Okay. Showed that photo of the glare glands in an earlier slide. Those whitish structures on the uropods or the underside of the tail fan and on the abdomen of the female. The female uses those glands to secrete this very sticky substance that we call glare. And she'll extrude that glare and move it to the back side or the back end of her walking legs and the underside of the abdomen. Basically, what she's doing is creating a pocket with that glare. And that glare is able to remove some water from that pocket. And then at this point, she releases the gametes from the base of her eggs, from the third pair of walking legs. Remember those gonopores up there? And will release that viable sperm that's stored in that annulus ventralis. And they'll excrete both of those gametes, and it'll move into this pocket, this glare pocket. Fertilization of the gametes occurs, fertilization of the eggs occurs, I should say, and eventually, within a couple of hours, that glare starts to dissolve and form into sticky abyssal threads. And those threads then are what allow the eggs to become attached to those swimmerets or pleopods. You can see here on the right side, on the underside of the abdomen. Anyone has picked up an ovigerous female, or what we call in berry, those eggs are stuck on there fairly well. You can put a little bit of pressure with forceps and pull them off. It's not like a super glue by any means, but it does hold them in place much better and allows the females to carry them much easier when she curls that abdomen up and around that cluster of eggs. All right. There's three stages or molts once those eggs hatch. Here's an image at the top center that shows, it's hard to say. I don't know if that's stage one or stage two. I'd guess maybe stage two because you can see start to see more shape in the individuals. But the young are attached by that abyssal or telson thread, which will, after they've hatched, that thread will eventually disappear by the end of stage one. They'll go molt into stage two, where they'll stay attached by small hooks on the chelipeds or the claws. They will, in some cases, detach from those swimmerets or pleopods on the underside of the abdomen to feed. And then will come back, and then after a stage three molt, the juvenile will leave the female. This whole process lasts, again, this is somewhat speculative, but most people think between 10 days and 2 weeks at the most for these stages, the eggs to hatch, these three molts to happen, and the juveniles to detach and leave. Again, most of this has been done with aquarium studies in the laboratory. Okay. Again, touching on that direct development, these things are considered juveniles or immatures, not larvae. When they do detach finally after stage three, all appendages are complete except the uropods. Again, uropods is that tail fan back here, not fully developed, and the first pleopods, i.e. the gonopods, or the gonopods in males aren't fully developed. Most populations of crayfish in the Midwest, you can normally, if you collect enough individuals, just talking about age classes, we'll get into longevity. Normally, you'll see 2 to 3 age classes. It's just an image showing that size difference. Most streams, when you sample, given that there's several weeks to a month or two when eggs are hatched and juveniles are released, you'll see some size variation maybe in the age one, but compared to an age two, a much larger individual, you can generally make that out. And of course, this slide is not showing age 0 individuals, which would be at least a half to a third the size of these age 1 individuals, much, much smaller. All right, so I titled this presentation Midwestern Crayfishes because I had the feel most folks would be from the Midwest. And so talking about a generalized Midwestern life history strategy, as you move further south in the US, you do see a fair amount of variation in that timing, life history, reproductive strategy. For example, some genera reproduce year-round. The genus Cambarellus is the most noteworthy. Those are the dwarf crayfishes. There are 2 species in that genus that occur in extreme southern Illinois. But I'm not sure if I don't know if we have enough data to say that reproduction is going on year round in the extreme southern tip of Illinois. But as you go farther south, talking about Alabama, Mississippi, you will see evidence of reproduction, I'll say, in most months. And some of the genera, Faxonius and Cambris, for example, too, that we have in the Midwest, you will see Form 1 males, fall to spring, which we see here. So similar there, but you can see the release of juveniles a little bit earlier in the year, from late winter, like February, into early summer months. So a longer release of juveniles in some of these genera farther south. Probably the best example of this variation in life history is in the genus Procambarus, which we have red swamp crayfish, white river crayfish here in the Midwest, although I'm sure they reproduce more typically in that seasonal pattern we've talked about. some species, not all, but some species of progambers in Alabama are found, ovigerous females are found February to October. I would guess that they're probably there year round. The reason we don't have this is based on field work and collections that are in museums. There's just not a lot of people out collecting in November, December, January, even in the far south. So we're probably missing some signal over those winter months. But if you find a female carrying um, eggs or hatched eggs still attached in August and September or July, you know that there's at least two periods of reproduction, typical spring and even over the summer into the fall. So, all right, growth and longevity, checking on time. Okay, so back to the molting aspect in life history, these things are arthropods, right? And so with an exoskeleton, they need to molt as they increase in body size. Um, what that confusing circular figure that I had early on that showed that generalized life history pattern did not show was that there's a lot of molting that first year through that first summer as those detached juveniles or immatures start growing. They're growing pretty fast over that summer and early fall months, and they might be molting every 3 weeks, once a month, something in that ballpark. Um, Generally, there's an increase in 5 to 10% in body size per molt. Again, as arthropods, they molt their entire lives, but going back to that circular figure, once you get into like year one, the males are only probably molting twice a year, and females too. Females will molt after releasing juveniles. It's an important component, and I want to talk about the importance of gastroliths. This is another really unique aspect of crayfish life history and biology. So over here on the right, there's images of what are called gastroliths, and this is a really cool strategy. Again, back to this bet hedging that we see possibly in females keeping viable sperm over a much protracted period. As crayfish get ready to molt, they start to break down the calcium carbonate in their exoskeletons and consolidate that calcium carbonate into these structures called gastroliths. These are found in the stomach. And what these gastroliths, they increase in size as that individual gets ready to molt. The individual then will molt. And what these gastroliths allow is you've basically stored up a little of that calcium carbonate as a jump start to start hardening up your exoskeleton. So as soon as the individual will molt, that calcium carbonate that's stored in these gastroliths will then be moved to probably the most important structures as you start to harden up, and that is your mandibles and the tips of your walking legs. You want those, you're going to need those structures right away to start feeding and start moving. So these gastroliths, this kind of, I don't want to say emergency, but this reserve of calcium carbonate allows that crayfish right after it's molted to start hardening up some of those important structures. You find these things. These are individuals that were sent to me by fisheries biologists that find these in the stomachs of fish. Fish love softy crayfish right after they've molted. And this is all you find sometimes in fish stomachs are gastroliths. If anyone's kept crayfish in an aquarium and one of the crayfish in the tank molts, you may come back 2 or 3 days later. If it happens on a Friday evening, you might come back on Monday morning and just find these structures on the bottom of your tank. The other individual will just totally consume that fresh mold. All right, longevity. It is hard to determine age of crayfish. There's no hard structures like bone structures like otoliths to age crayfish. You can look at length, size, frequency distributions and take a guess. There's some data that says there's some aging that goes on in the eye stalk or eye tissue. Um, our best guess is open water species, probably 2 to 3 years, maybe 4 here in the Midwest. And I would speculate that some of the larger burrowers that we have in the Midwest may go 5 years. Some of the larger species that are in the Pacific Northwest that are in that other family, the Astacidae, there's some data that suggests those will live 7 to 8 years. All right, transitioning to feeding. Um, Crayfishes historically were treated as general omnivores and scavengers. And that still may be partially true, but as more folks have started to work on crayfish, some of those that kind of foundational theory has started to shift a little bit, at least with a handful of crayfish species. So again, generally considered omnivore scavengers. Walter Momot in 95 published the seminal work on Virulus in Canada and concluded that they were obligate carnivores first, feeding on macroinverts, and then became facultative omnivores and scavengers later in life. But not too much longer after that, some work in Scandinavia, again, or different family, but did show that some of the Astacid species did just the opposite. They started as omnivores or scavengers or herbivores early in life as their juveniles or age one individuals because that food source was made more abundant. And then as they aged into older individuals, they switched to become primarily carnivorous. All right. There's also data that shows they may become more carnivorous with age and body size here in the US. This is a project one of my graduate students did years ago that showed differences in trophic position of crayfishes at the same location. Trophic position here on the y-axis, and you can just see these symbols are different species. Barbicamberus cornutus, or this is Barbicamberus simmonsii in the photo, but the two species in that genus are two of the largest species in North America. You can see that generally at sites, these are 3 different sites, top left, top right, bottom left, show that in all cases, the Barbicamberus species were feeding at a much higher trophic level, i.e. they're spending more time eating higher protein matter, either fish, other crayfish, or invertebrates. So there is some variation in feeding that have been documented. All right, this is way out of focus. I'm not sure why. This is just another study with 2 Midwestern species, rusticus and propinquus, that show that this is percent of the diet. That does show that between fish, invertebrates, and crayfish, adding these 3 numbers, the point of this paper was showing that crayfish do eat fishes. You used to look at old energy, food pyramids, and it would always show fish above crayfish. This is just showing that it can go the other way, where crayfish do eat some fish, but between invertebrates, other invertebrates, insects, fish, and crayfish, maybe 40% of the diet could be composed of current other animals. So, all right, figuring out what crayfish eat other than using stable isotopes can get difficult because of the gastric mill that we find in crayfish. Crayfish have 2 stomachs. They have a cardiac stomach and a pyloric stomach. Cardiac stomach is in front, pyloric's in the back, but as you move from the cardiac to the pyloric stomach, that food material goes through a gastric mill. These are all hardened, sclerotized, I'll say teeth that are used to grind up that food matter. So unless you examine a crayfish before that food moves through the gastric mill, it gets extremely difficult to identify food matter back here in the pyloric stomach because it's ground up so fine. So looking at diet studies in crayfish is complicated by this. It's all about finding them at the right time. Once that food gets past that gastric mill, it's extremely difficult to identify what you're looking at. So another complicating factor for diet. kind of talking about, just finishing up here, talking about where crayfish fit in food webs. Most of you already know that there's an old a lot of things in aquatic ecosystem eat crayfish. Every fish, birds, reptiles, amphibians. I think these were numbers we put on this workshop years ago, and I'm sure these numbers are much higher now. Over 25 fish, 18 herps. I don't have a number for birds, but I'm guessing there's at least 50 different bird species that have documented crayfish consumption. So crayfish eat everything, and everything eat crayfish is the saying that you can hear, probably partially true. Um, in that role, crayfish as recyclers and mid-trophic level consumers, although that slide I showed earlier showed that they might be top level consumers. So again, it's not a one size fits all, but certainly as omnivores and scavengers, there is a fair amount of work that shows crayfish function in that important mid-level consumer that will serve as that link between primary producers and animal tissue. Okay, moving that plant matter up to top level centrarchid predators in any aquatic ecosystem. Very important in that role. Um, Often the greatest invertebrate biomass, obviously given the large body size and the abundance of crayfish that occur in some sites are often more than 50% of invertebrate biomass. Um, Impacts of perturbations in aquatic ecosystems. There is some work that show that heavy metal toxicity, i.e. cadmium, a lot of this was done in the Missouri Ozarks, um, can be detrimental to crayfish populations, but the impacts of other perturbations are not well understood. I will say this, generally, crayfish are regarded as a tolerant organism in that role of theirs as mid-trophic level consumers. It may not be true for all species, but considering the other kind of peer groups in freshwater ecosystems here in the Midwest, i.e. unionid mussels, of course, fishes, crayfishes will generally be a little bit more tolerant of disturbance. Um, there's also some symbiotic relationships that we see with crayfishes. I don't have time to go into detail for those, but branchiodellid worms, for example, are very well known to inhabit crayfish, sometimes multiple species of branchiodelids in multiple locations on crayfishes. Ostracods are known to attach to crayfishes. And then another one here in the Midwest that maybe not as well known are carixids. These are water boatmen, a very, very common invertebrate insect group that we have here in the Midwest. And they will lay their eggs on crayfish. They will lay their eggs on vegetation, but they also are known to lay them on crayfish. This is an image of a museum specimen, and this individual is just covered with really, really tiny eggs for carixids. So if you ever see an individual, this happens frequently. Someone will be sampling. Normally, it's more stagnant water, but you'll pull out an individual and it'll be covered with these small little, they look like small little eggs, and not know what they're looking at. Those are carixid eggs. Um, we'll just move close quickly here. It looks like we're getting close to running out of time on ecological categories of crayfish. Again, this is old hat for a lot of the folks that are on this webinar. Generally, two main ecological categories of crayfish: Apygian surface-dwelling species and Hypygian subsurface species, cave dwellers and burrowers. As most of you know, surface dwelling crayfish will occur in almost every type of aquatic habitat. Sloughs, swamps, creeks, rivers, large navigable rivers. If there's water, generally you're going to find them. I will say permanent water. However, those other ephemerally flooded habitats or subsurface waters like caves, um, flooded roadside ditches here, you see in the lower left, you will see the other major ecological category, which includes two, burrowers and cave dwellers. Burrowers, most of you are familiar with seeing mud mounds in ephemerally flooded habitats, but here's the sticker. All crayfish can burrow, and they've generally been classified into three categories: tertiary, secondary, and primary. These are some of the genera that we have around the Midwest. Some of these down here in the primary burrows, Distocambarus, Creasorinus, or sorry, Phallicambarus now, Distocambarus don't occur here. Okay. What's the difference between these three categories? Primary and secondary are generally lumped together they're generally called burrowers. Primary burrowers, this is a classic illustration by Hobbs in 81. Primary burrowers normally will have a direct connection to a permanent water body. Sorry, no direct connection. Misspoke there. As you can see, the red lines here, these primary burrowers, they can be very extensive, very deep systems of tunnels and cavities. And in this illustration at the bottom, you can see that there is no direct connection to a permanent water body that you see in secondary burrowers. So if we look at this secondary burrower here, there is this tunnel that's going over to this permanent body of water. And that, in theory, is the main difference between primary and secondary burrowing species. The chimney structure, the burrow opening between the two look identical. You wouldn't be able to distinguish if it's a primary or secondary just based on burrow morphology in most cases. You probably get an idea of a position where you find that chimney or opening or that mud mound. It would give you an idea if it's a primary or secondary. Primary burrows can go very deep. Got this note here at the bottom, up to 3 meters. That's an incredible depth for a smaller invertebrate organism to dig a hole that deep. Down to the water table, just some stuff here, multiple passages, large chambers. They will plug those burrows in dry or cold periods, and chimneys vary in size and structure. Chimneys are not diagnostic for species in most cases. Tertiary burrowers are then everything else. This is just a simple burrow in the bottom of a stream channel over here to the right. And tertiary burrowing conditions will happen in winter to get below the frost line in some cases. Sometimes you'll find females that are getting ready to expel those gametes into that glare chamber and egg brooding. They'll sequester themselves in out of the way areas, under woody debris, under logs, under rocks, or will sometimes build a very shallow burrow to brood those eggs in until they're hatched and ready to go. And then, of course, during drought conditions, crayfishes, all crayfishes will have that ability to dig this very simple, shallow burrow to get down into moist soil, keep their gills wet. Cave dwellers, not a whole lot to say here. We don't really have any in the Midwest, but there are two different types. Stygophilic and stygobitic. One is those species, I'll take that back. One species that does live around and in caves, but normally only in that twilight zone of caves. They don't get as far back. They do have pigment and they do have pigment in their eyes. Cambrus tenebrosus is a perfect example of that. That's a species that occurs here in east central Illinois, in Indiana, and down into southern Illinois. It's normally associated with spring outflows or cave openings. And then we got the two true cave dwellers that are stygobitic that have lost their pigment and lost pigment in the eyes. Southern Indiana does have cave dwellers, so I guess I should correct myself. Indiana, we generally consider a Midwest state. All right, that is it. Looks like we're fairly close to timing. I would be happy to answer any questions, and if I can't answer them, I'm going to punt it over to some of the other experts that are on the webinar.
Great. Thank you so much, Chris. Currently in the chat and in the Q&A box, we don't have any questions. Um, but I'd like to ask a few kind of while our attendees are potentially forming theirs.
Okay.
I'm particularly interested in gastroliths. I think the production of them is just so fascinating.
Yeah.
In grad school, I used to collect them in jars and kind of shake them around like
Mhm.
Skittles. It was really funny. But I was just wondering, how does the calcium availability in the environment where the crayfish exists, how does that influence gastrolith formation, and the molting success? So I guess like do all crayfish produce gastroliths when in areas with low calcium?
Well, I don't know for sure, but my guess is going to be yes, because if there has to be enough calcium in the environment to form the exoskeleton to begin with. And I have been to locations that have really, really low dissolved salt levels, and you just don't find a crayfish there,
Mm.
even though the stream looks fine. So my guess is, unless somebody else knows more than I, is that if there's enough calcium in that water to form an exoskeleton, there's going to be enough to break it back down and form those gastroliths.
Gotcha. Thank you. Still see. Oh, okay. We got a question in the chat. A few of them. So this one's from Patrick Siula. With projected climate change scenarios, do we think we might find reproductive strategy changes, such as potentially spawning more than once per year?
Yeah, I'm gonna stop. Yeah, I'll leave it. Um, Possibly. Yeah. There's been a little bit of work on projected distributions of crayfishes as a result of temperature change. And the results of that modeling work showed that there are still other more important factors for modeling distributions of crayfishes other than temperature, which suggests that, again, getting back to that comment about crayfishes generally being a little bit more tolerant of habitat change, um, I I'm not sure. It would pure speculation here. I think it would take significant temperature change of more than 3 or 4 degrees before you start to see a shift in reproductive strategies here in the Midwest. Remember that reproductive time frame is still even pretty protracted here in the Midwest. You can see copulation going on from early September into November, and you may even see it in the early spring months. So it's almost like they've already built in some flexibility to temperature change. Um, you don't see really hard and fast, and that's just based on this length of reproductive activities. You don't see kind of a real sensitivity to exact temperature change in crayfish. So my speculative answer to that question is it would take some significant changes to see some shifting because they already seem to have some plasticity with response to spawning activities.
Great, thank you. Another question in the chat asks, are certain species more likely to be found in lakes? Are there only certain species to be more likely present in lakes?
Um, I would say, at least here in the Midwest, the answer to that question is no, because all of the natural lakes that we have are occupied by species that also occur in more lotic environments. So there aren't any lake specialists, lake habitat specialists here in the upper Midwest, where you'll see a species just in lakes. You might find higher population levels of some species, but I think that's more going to be an influence of um, substrate type. Crayfishes need to have something to hide under, like rocks of a given size or woody debris to get away from other predators, fish predators mainly. And so if there's a lake that has an ideal substrate of larger, medium-sized rock that allow crayfish to get under them, you will see larger populations of a certain species in a lake versus a stream, depending on bottom substrate. So um, natural lakes here in the Midwest, no, we don't really see lake specialists that are only found in lakes. As you get farther south and you get more into um, sloughs and swamps that are more lentic conditions, yes, you will definitely see. And when I say farther south, I'm talking about probably farther getting down into Kentucky, Tennessee, and then down into the deep south in the Gulf states. There are definitely species that you will only encounter in those oxbow type lakes and more slough or swamp type habitat. So uh, I hope I answered the question.
I think you did.
Okay.
Um, we have a couple more questions in the chat, and then we'll have to wrap up since it's kind of the top of the hour. Another question in the chat asks, when do adult Faxonius molt back to F2 in the Midwest?
Well, back to F2. Um, I'd say May to June in most cases. If you're out collecting in the summer, most of the males you will encounter are Form 2. So again, you know, most of these dates when you see molts or transitions from one form to the other aren't near as fixed as like reproductive activity in some species of fish. Some darters you'll see, they're only spawning at a certain temperature range in May to mid-June. A little bit more protracted periods of these activities in crayfish, but generally as that kind of weird circular life history strategy showed, from my experience, it's going to be late Mayish into mid-June usually, but that's a broad generalization. You'll see that molt back into Form 2.
Great, thank you.
Mhm.
And then the last question, is there a general understanding of surface water crayfish behavior during winter? Do they burrow in benthos and reduce overall activity, or is it varied between species?
Yeah, I'm not sure I can address the vary between species because there's just not enough data to address that. But generally, yes, there is a slowing down of activity and kind of, again, since fish are still somewhat active, generally sequestering themselves under some kind of substrate like woody debris or under larger size rock or slab rock or things like that, and just not a lot of activity. But given the severity of our winters and the challenge of doing that kind of work, I've just not seen a whole lot of peer-reviewed literature. Somebody else on the webinar, if they've seen some stuff that looks at behavior and during those winter months. It's just there's not a whole lot of data out there to know what they're doing, just other than a lack of activity level given the temperature.
All right. With that, we are going to have to unfortunately end the call. If anyone has any additional questions, can they reach out to you, Chris?
Yeah, absolutely. Yep, for sure.
Okay. I pasted Chris's email in the chat, so if you have any additional questions, feel free to forward them to him. Um, a big thank you again, Chris, for taking the time and giving us a really great presentation about the ecology and life history of crayfishes. I feel like that's a very a needed thing to learn about right now.
Yeah.
And thank you everyone who joined us today. Hope you all have a great rest of your week, and a happy holiday season. Thank you so much.
Yep. Thanks all. Let me know. Send me an email if you got a question. Happy to answer.